Well, thank you everyone for giving me the opportunity really to come here and tell you about my work. So I'm Jimena, I'm a postdoc in Sergio Pascal's lab at Stanford University. And today I want to tell you about some of our efforts to generate reliable human cellular models to study some of the mechanisms that underlying neurodevelopmental disorders. And so first I will tell you about the model itself that we generated, and then I have some preliminary data on PCDH, although not that much, unfortunately. But anyway, I think I was very lucky today because this morning I had great introductions to what I'm going to talk about today. So in the lab, we are interested in the development of the cortex. And as we heard today, the cortex is the outermost layer of cells in the brain. And it's important for cognitive function, for sophisticated motor control. And we also heard today that for proper functioning of the cortex, we need a correct balance of excitation and inhibition. And this excitation and inhibition balance relies on the proper assembly of excitatory neurons and inhibitory neurons into a functional network in the cortex. But what you need to know is that these two different neurons are actually born in very different parts of the brain during development. So here you have a section of the developing developing forebrain. And what you see is that while excitatory neurons, so they're here in red, they're born in the cortex itself, interneurons, they're actually born in this ventral part of the forebrain, which is called subpallium. And these interneurons need to migrate very, very long distances over several weeks during development to reach the cortex. And once they reach the cortex, they need to integrate and they, in order to create these functional networks. And so now we know, or we're starting to get an idea that any disturbances to this process can actually lead to excitation to inhibition balances and therefore possibly contribute to epilepsy and other disorders like autism spectrum disorders. And so for us, it was important to generate a human model that would actually recapitulate some of these complex cellular processes that take place during development. And so for that, and again, I had some introduction to this this morning, but we used induced pluripotent stem cells. And with this fairly recent technology, what we can do is actually get skin cells or fibroblasts from any individual, and then we can reprogram them into an embryonic like state that is pluripotent that enable us to push these cells into any fate that we need based on the developmental cues that we know from the development. And so once we have these pluripotent stem cells, which I will call iPSCs, we can grow them in culture. And then when we want to differentiate them into neurons, what we can do is we can lift them from their plate, and we can put them on a plate that is low attachment, which means that they're going to just grow in 3D as little spheres. And then we give them certain growth factors to push them towards a specific fate. And then in the end, we end up with 3D neural brain spheroids. And so this is what they look like early on. So this is day 13. And you can see these rosettes or progenitor. Hopefully you can see these progenitor areas that were mentioned before as well. They're quite small initially, but as they grow, they actually grow considerably in size. But at the point that by day 60, they're comparable to an embryonic mouse brain. And so because these are floating spheres, the process is actually quite scalable, and it means that we can keep them in culture for a very long time. So these that I'm holding here, I think are about 150 days old, but we have actually managed to keep these cells in culture for up to 700 days. And so we can generate 3D brain structures. And again, I'm going to bring you back to development to remind you that the cells we're interested in are born in different parts of the brain. And so we thought, if during development they're born in different parts, then why not generate them separately and then put them together to model the processes that take place during the development. And so that's what we did. And again, we're going to start with iPSCs, or pluripotent stem cells. We're going to lift them off the plates. We're going to give them a number of factors that are going to push them towards the neural fate. And then we're either going to give them patterning cues that are going to drive them towards a cortical fate, so the excitatory fate, or we're going to give them other cues that is going to drive them towards a subpallial or ventral fate that it's going to generate the interneurons. And so we just wanted to make sure that the cells, the spheres that we generated actually are the regions that we're interested in with the relevant cell types. And so for that, we actually went on to do single-cell RNA sequencing. And so for that, we can separately dissociate cells of either the cortical or the subpallial fate, and then do single-cell RNA sequencing. And so here what I'm plotting is each dot is a cell. We have about almost 1,200 cells. And what you see is gene expression in cells, and what you expect is cells have similar gene expression, then they're going to be closer together in this plot. And that's what you see. So cortical cells, which are the purple ones, are on the top, and subpallial cells are on the bottom, and you see that they cluster separately. And then within these clusters, we actually see subclusters of cells that we would expect for these brain regions. So for example, we have a group of glutamatergic neurons that express excitatory markers like V-GLUT. We have two groups of intermediate progenitors that express markers like TBR2, which is a progenitor marker. And we also have a group of dorsal progenitors, this here, the orange one, that include both ventral and outer radioglia that we also heard this morning about. And that is in the cortical cluster. In the subpallial cluster, we actually see GABAergic neurons, which are the inhibitory neurons, and we see ventral progenitors again. And at the age that we are doing this, which is day 100, we actually also have astrocytes, and we know that astrocytes are important for neuronal function. They help the neurons, let's say, to survive and to function. And so we have also astrocytes in these spheres, which is very important. And interestingly, the astrocytes from both the cortical and subpallial sites clustered together. And so we have the cells of interest in these two region-specific spheres. Then we went on to just look inside and make sure that they look what we expect them to look like. And so in the cortical spheres, we have, as again we saw this morning, a ventricular zone area where the progenitors are, with deep layer neurons expressing CTIP2 and superficial layer neurons expressing SAPI2. We can also transcriptionally compare our spheroids at different time points with the developing human brain. And we see that, it's not very clear, but at days 50 and 75, actually, our spheres are correlating transcriptionally to the mid to late developing brain. Now, in the subpallial spheroids, we see early on, so this is day 25, we see the progenitor marker NKX 2.1, and later on we see expression of GABA and the GABA synthesizing enzyme GAD67, and GABA is the main inhibitory neurotransmitter in the brain. And we also see expression of the main subtypes of interneuron markers like somatostatin, calretinin, or carbindin. So again, we have our regions of interest. We have our excitatory neurons that are born on the dorsal part of the brain. We have our inhibitory neurons that are born on the ventral part of the brain. And then the next step is for us to put them together to model the migration of the interneurons. So that's what we do. And because we're generating them together, we're generating separately, we can actually label cells from either side separately so that we can visualize them over time. And so in this case, we're using a viral reporter that marks neurons in the subpallial side, so the interneurons. And just briefly, this is what it actually looks like. So these are cortical and subpallial spheroids that are then put together, and after a couple of days, they just form one structure. And if we look at the expression of this reporter over time, what we see is that indeed interneurons move from the subpallial side to the cortical side over time. And this seems to be specific to the subpallium, because if we label the cortex with another reporter, we don't see cells moving to the subpallial, cortical cells moving to the subpallial side. And so we can do live imaging and follow this migration over time. And so this is a video of what that looks like. So here we have the subpallium side on the bottom, the cortical side on the top. This is a video of about 12 hours of some spheres that have been assembled for about 2 weeks. And you can see that interneurons from the subpallium have moved to the pallial side, because again, we've labeled these cells before we actually put them together. And so interneurons have this very particular way of moving. So they move in this saltatory pattern. So I'm going to zoom in so that you can see this, hopefully, this movement. So what you'll see is that they extend a leading process that basically is like looking where to go. And once it decides where it wants to go, it will have a transient swelling of the soma and then the movement of the nucleus. And so it looks like the cells are jumping when they're moving. And this is what this nuclear movement looks like. This is the nucleus, and that's the rest of the soma, and that's called nucleokinesis. So hopefully you can see some of these cells, like this one here. You can see it jumping. So if we actually look at these assembled spheroids after about 4 weeks, you can see that a lot of interneurons have moved to the cortical side, and this is a 3D reconstruction after this iDisco, which clears tissue. So this is very nice, but of course, how do we know that this is relevant and this is what happens in vivo? And the only way for us to actually test that was to check primary tissue. And so that's what we did. We got human forebrain tissue, and labeled it with the same reporter that we use for the spheroids, and we imaged it in the same setup that we do. And so here you have two cells. This cell is from an assembled spheroid, and this cell is from the human primary tissue. And you can see they are very, very similar. And of course, we can quantify all of this movement. And the kind of things that we look for are the size. How is the size of our neurons compared to the size of primary neurons? And so for that, we looked at the branch to soma ratio. We can also look at the number of saltations. So how many jumps do they do over time? And how much they move every time they jump. What's the saltation length? And of course, the overall speed of the movement. And so when we do that, you can see, so in the darker green is the assembled spheroids interneurons, and in the lighter green is the primary neurons. And you see that they are almost identical in their migration dynamics. Now, this movement has actually been characterized in mice extensively before. And so we wonder, how does this compare to mouse interneural migration? And so again, we did the same. We got mouse slice cultures at E18 and compared the movement to our human interneurons. And what you can hopefully see is that mouse interneurons are actually about half the size of human interneurons. And while they actually do more saltations over time, and therefore their speed is much higher, the length of each saltation is the same as humans. So that's that one here. So this is exciting because it tells us that we have a model that in which we can ask specific questions about the migration of interneurons in a human context. And so the next thing, of course, is to ask, can we use this to model disease? And so for that, we first started with patient cells that were from patients with Timothy syndrome. So Timothy syndrome is a disorder that is highly penetrant, is monogenic, is characterized by epilepsy and autism spectrum disorder, as well as cardiac arrhythmias and sudden cardiac death. And so it's caused by missense mutations in a subunit of a L-type calcium channel, CAV1.2. And what these mutations do, basically, they decrease, they have a delay in activation in the channel. And so what that means is that once they are depolarized, they take longer to close the channel, and so that means they have more calcium in the cell. And that's shown here with KCl depolarization. And we know that calcium is important for numerous processes during development and in general, and it's important for migration. So we thought it would be a good model to pursue to try to see if we could use this system for disease modeling. And so I'm going to show you a video, and these are, again, a control cell and a cell that has been derived with patient CS cells. And again, you're going to be looking for the same characteristics that I showed before, so the saltation length, how many saltations they do. And so if I play it, hopefully what you see is that although the TS cells do more saltations over time, their saltations are smaller, which means that they're less efficient at moving. And so this is characterized here. The gray is the control cells, and the green is the TS cells. And here it's again quantified. We can see that the number of saltations that they do is significantly higher compared to controls, but the length of the saltations is much reduced. And because we can separately generate these spheres, we can actually create hybrid systems where we can put a control cortical and a TS subpaleo to try to assess the cell autonomy of the phenotype. And so when we do that, we can see that this is a cell autonomous effect. So the interneurons, the defect is not on the cortex, it's the interneurons themselves that have the phenotype, because the phenotype doesn't change when we do that. And of course, we can then try to rescue this phenotype. And we did this with nimodipine. So nimodipine is a calcium channel blocker. And so when we do that, you can see that, so here we have the control cells, and this is the saltation length. Here we have the TS cells. And while in the normal baseline, we have the TS cells have much shorter saltations. When we add this drug, this is rescue, and the number of the saltation length is back to a control level. So this is exciting, and it tells us that we can use the system to model disease, or at least to ask specific questions about some of these processes that are taking place during development. And so, of course, the next question that we ask is, can we use these cultures and assembloids to model PCDH19 epilepsy? So we heard today a lot about the possible functions of PCDH19 epilepsy. And so I won't go into detail about it, but from what we heard today and from the literature, what we think is that PCDH is involved in cell-cell interaction, migration, and synapse formation. And so these are things that we can actually model with our system. So we can separately specify cortical and subpaleo spheroids from PCDH19 patients. We can then assemble them together to study the migration and ask whether there are any defects in the migration on these cells. And we can also look at the integration and synapse formation. So I haven't shown you here today. Sorry about that. There we go. So I didn't show you this today, but we have actually also shown that once the interneurons move on to the other side, so they move from the subpallium to the cortex, and when they arrive to the cortex, they change morphology and they start having many more branches. They start upregulating expressions of genes that are linked to maturation of the interneurons, and they also start changing the electrophysiological properties. So that also tells us that we can actually look at the integration of the interneurons into the system, the formation of synapses, and the formation of early cortical networks. So these are still very early days. As I said, I don't have that much data, but I want to show you what I do have for now. So studying PCDH19 gets very complicated very quickly once you introduce X chromosome inactivation, as we have heard before. So what we decided to do to start with was put X chromosome inactivation to the side and just ask if we generate spheres from control and PCDH19 cells, do we see any phenotypes? So that's what we did, and for now I'm just showing you cortical spheroids. We generated spheres from 2 control females and 3 PCDH19 cells were actually derived from 2 patients. And then we collected the spheres every 25 days for 100 days. And then went on to do whole genome RNA sequencing. And so here, again, I'm showing you, hopefully you can see, very light, so here, this is a plot that I'm basically showing the relationship between all these samples in space. And so you can see that first that these different samples, so we have the control is the open circle, and PCDH is the filled one, and then the different colors are the ages. And so we can see that these samples are being separated by age. So we have here the youngest ones, and then as time goes by, they move that way. But we can also see that there's a separation between the control samples and the PCDH19 samples down here. So that's exciting to know that we can see a difference between these 2 genotypes. And then we asked, well, what's different? And we did differential gene expression, and we found a number of genes that were either downregulated or upregulated in PCDH19. And then we went on to do gene ontology analysis, which basically highlights important pathways that are enriched in these genes that are differentially expressed. And so it was very exciting to find that the first term that comes up is actually homophilic cell adhesion, which I guess is not unexpected. But then the other terms that come up are also very interesting. So we have synapse assembly and positive regulation of cell motility. So again, this is very preliminary, and we still have a lot of samples that we want to add, but it's telling us that we can use this system to study PCDH19. And so the next thing that I will be doing, and I really just started this, so this is the first video that I've made, but I will be generating cortical and subpallial spheroids from control in PCDH19 patients and studying the migration. And I can't really say anything about a phenotype yet, but hopefully I will have this data soon and I'll be able to share it with you. So really, I want to say 3 main things that you should take home about this talk. So one is that using human-induced pluripotent stem cells, we can pattern in 3D spheres into specific fates, and we can use these region-specific spheroids to model complex cellular interactions that take place during development. And importantly, that we can use these spheres to model disease, and hopefully I can show you some data on that on PCDH soon. So I just have left to thank everyone in the lab. So let's see if we find Sergio. That's Sergio, he's my mentor. And yeah, so he's been great. He's really hardworking. And well, everyone else in the lab is very hardworking as well. We have a lot of collaborations. Really, these projects are highly collaborative. So Aaron and Dan in UCLA have helped us with the PCDH19 RNA-seq. Chris, I haven't shown you here, but Chris has done all of the electrophysiological characterization in these assembloids. And yeah, we have help with single cell, with the mouse experiments. Yeah, so and thank you very much for listening.
So, questions for Jimena? Got to get this started.
Yes, thank you. It's really lovely work to look at it and how long you can culture these, oids, because there are so many other assembloids, spheroids. You mentioned that you have some for several hundred days. What are you trying to look there? This is perhaps not necessarily related, but are they increasing in complexity over such a long period of time? And it is the right direction, or it's just a runaway train and maybe it's not going to be point, but we can't do a 700-day experiment. Well, that would be difficult.
Yes, thank you. That's a very good point. So most of the experiments we do are up to day 200, I would say. But we've been trying to collect all these time points until day 700, and we've been doing RNA-seq and ATAC-seq and all the seqs to try to compare that to the human fetal brain and try to see how does it compare. Do they do all these things? Of course, they probably won't. They're missing a lot of cues. They're missing a lot of their partners. They need connections. They need other parts of the brain. So we're definitely missing a lot of it. What we do know is that astrocytes, at least, up and I think it's around 9 months, they start to become more postnatal looking. So maybe astrocytes don't need that much like neurons do.
What a lovely system that was, and the movies are fantastic. I can't wait to see what happens when you put some mutant and wild type oids together. With the last experiment you showed us with the predicate here in 19 mutant mixed cultures, I think, so they're from heterozygotes, is that correct? Yep. So did you look at the expression of predicate here in 19 in those bulk seq data, was it? Not single cell seq, but bulk RNA seq. So what was happening to the levels of predicate here in 19 in those?
Okay, so yeah, as I didn't go into the mosaicism, or we actually believe that the X chromosome inactivation is very dynamic, that it changes over time. There's even reports of PCDH19 escaping inactivation. So we don't really know how to tackle that yet. But what I can say is that we've seen these, at least control spheres, show this non-skewed pattern of inactivation. And regarding PCDH19 expression, interestingly, it wasn't changed in the RNA-seq. So the first term, the cell addition term that comes up, is basically due to a lot of clustered protocadherins that are misregulated, although PCDH19 wasn't. So yes, something that we need to explore. I mean, I wouldn't be surprised that cells try to compensate for PCDH19 being lost in some cells, and so maybe they're trying to, express more. But then I guess that doesn't change the fact that it's not there, and the adhesion is going to be disrupted.
That was lovely. Can you use this model for functional studies instead of, let's say, using GFP, using GCAMP or other stuff? I mean, that would be really interesting.
Yes, thank you. So yes, the answer is yes. I didn't show any of this, but we can do, calcium imaging. We can use GCAMP. We can do slice recordings, to do whole cell patch clamping. We can use MEAs to look at networks. So yes, eventually that's the, I guess, ideal experiment. We would have cells that we know are either expressing or not expressing PCDH, and we can look at the network dynamics to see if that's misregulated. So the answer is yes, we can do it. I haven't done it yet.
And this may be a silly question, but can you do neurophysiologic recordings on the OIDs?
So neurophysiological recordings like, so we can use MEAs. We can like look at like cells. You mean like EEG kind of thing? Yeah, no, I don't, at least we haven't tried.